Hello and welcome to SIMPIC 2024 conference. We are delighted to have you here joining us in the theme of technology trends in the microbiological world. Today, we have the privilege of hosting presentations from Dr.Thidathip Wongsurawat, Dr.Chayasith Uttamapinant, and Dr. Piyalitt Ittichaiwong. If you have any questions, feel free to ask them after each speaker concludes their talk.
Hello and welcome to the SIMPIC 2024 conference. We are delighted to have you here joining us in the theme of technological trends in the microbiology world. Today, we have the privilege of hosting presentations from Dr.Thidathip Wongsurawat, Dr. Chayasith Uttamapinant, and Dr. Piyalitt Ittichaiwong. If you have any questions, please feel free to ask them after each speaker concludes their talk.
Now we are waiting for the live session to get ready. Ladies and gentlemen, distinguished guests, and esteemed participants, a very warm welcome to SIMPIC 2024 conference. I am honored to stand before you as your master of ceremonies for this extraordinary event. Today, we'll embark on a journey into the ever-evolving landscape of the microbiological world, exploring the cutting-edge theme of technology trends in the microbiological world. Our distinguished speakers, Dr. Thidathip Wongsurawat from Siriraj Long-Read Lab, Head of Division of Medical Bioinformatics, Faculty of Medicine, Siriraj Hospital. Dr. Chayasith Uttamapinant, School of Biomolecular Science and Engineering, Vittayasiri Methi Institute of Science and Technology. Dr. Piyalitt Ittichaiwong, Siriraj Informatics and Data Innovation Center, Faculty of Medicine, Siriraj Hospital. They are here to share their invaluable insights and knowledge with us.
So, without further ado, let us delve into the wealth of information that awaits us today. Each speaker will present their expertise, and after each presentation, you're encouraged to engage with them by asking questions. Your curiosity and active participation will undoubtedly enrich our collective understanding. Once again, welcome to SIMPIC 2024 conference. May this conference be a source of inspiration, collaboration, and innovation as we explore the fascinating intersection of technology and microbiology. Let the journey begin.
Now, we would like to invite Dr.Thidathip Wongsurawat from Siriraj Long Read Lab, Division of Medical Bioinformatics, Faculty of Medicine, Siriraj Hospital, Mahidol University, to lecture on the topic, Long Read, the New Era of Sequencing Technology. Everyone please give her a warm welcome.
Thank you so much. So, welcome to new era, long read sequencing technology. You can just call me Tip. So now I am working here at Siriraj Hospital, and I have lab here in case you want to visit. So, and also I am an adjunct faculty at the University of Arkansas for Medical Science, USA, and also be a consultant of the Division of Global Health Protection, CDC, USA. Okay, so I started working here in 2021, moved from US to Thailand, and at the beginning we have two persons, wet lab as me, and bioinformatics, Dr. Pilun. And then I established a small lab called Siriraj Long Read Lab. And then we hire more bioinformaticians and wet lab people and bioinformaticians. And we need two more if you want to apply. So, and my lab is here at Siriraj Hospital. The core technologies that we use is Oxford nanopore technologies and bioinformatics. So before I move to long read technology, let me recap about the central dogma of biology. It is an explanation of the flow of genetic information from DNA makes RNA and RNA makes protein. And the focusing today is DNA level. So the next word is genome. Genome is the complete set of genetic information in an organism. You can see here, different organisms has different size of the genome. The virus has only maybe 170,000 base pair, but human, the genome size is huge compared to virus, right? And you can see here E. coli. The size of the genome is about 5 million. So this is the popular DNA sequencing platforms, how to study the DNA. This technology, we can use this technology for sequencing the DNA. And I show here 3 platforms, Illumina, PacBio, and Nanopore. So Illumina is a popular technology that's used for short read sequencing. What does it mean, short? It's been 100 to 300 base pairs. And 2 technologies here, PacBio and Nanopore, is long read sequencing technology. It's above 1,000 and sometimes can be million base pair. So, and Nature Methods published last year in January about these 2 technology. They call Method of the Year in 2022. But I put the price, you can see the price of the machine here, very different. So why we have to focus on long read sequencing technology? I want to show you a simple slide. Let's say in the database, you have E. coli and Salmonella. And then you use short read sequencing. It's like you have jigsaw, small jigsaw. And when you map this jigsaw to this database, what happened? So you can map to E. coli and Salmonella. It means that 100 to 300 base pair cannot tell you which species, right? But if you use long read technology, the jigsaw is bigger, and then it can map only one organism. So in this case, long-read technology can give you a better taxonomic classification. And it's not just the cartoon. People compare between short-read and long-read, and they found that nanopore or long-read sequencing technology provides superior performance compared to short-read sequencing. But why I focus on Oxford nanopore technologies? Because when I moved here as a new staff, no one invests 40 million baht for me, or about $1 million for me. Yeah, and I don't have space, as a new staff. We need space and the room that control the humidity if I use PacBio. And also you have to think about the maintenance cost. Yeah, when you get the grant, you can buy maybe big machine, but you have to think about next year. How can we pay for the insurance, for the warranty and many things, right? Aircon and something. So we have to think about that. That's why I moved to nanopore technologies. And what is nanopore? First, we start from nanoscale. You can see here human hair. The size of the human hair is about 80 to 100 nanometers. And DNA is only 2 nanometers. So nanoscale means 1 to 100 nanometers. So nanopore is a nanoscale hole, it's tiny hole. It can be biological nanopore, such as protein pores. It can be solid state, like graphene, or it can be hybrid between protein and synthetic material. For this company, the Oxford Nanopore Technologies, they use protein pore. And the size of the device, you can see here, is like a chocolate bar, the Snicker. And the device, we don't need to pay for the maintenance cost. It's $1,000 US. But the flow cell is quite expensive. The flow cell is a consumable item that $900. So when we zoom into the flow cell, it contains about 2,000 nanopore. And this is the cross-section of the nanopore. You can see when we turn on the minion or the device, some people call minion. Yeah. When we turn on the minion, and then the ionic current can pass through the pore. And the sensor can detect the current and plot the graph, like this. But when A, T, C, or G, or the DNA molecule comes to the pore, what happens? The ionic current cannot pass through smoothly. So the current drops, and the graph can show like this. Then when the DNA strand moves through the pore, ATCG has different sides. Yeah. So it can create pattern like this. And then we use bioinformatics to change that pattern to be the DNA sequence like this. So this is called base calling. And this is the video to show when we load DNA onto the flow cell, what happened. The double-strand DNA, and you can see the green color there is the helicase that can denature double-strand DNA to be a single-strand DNA and push the DNA into the pore. The speed is about 400 bases per second. This is the slow motion. Yeah. And you can imagine if we have 1,000 nanopores, you can get huge data from sequencing. And this technology, this one I copy from their website. They say that their technology can sequence anything by anyone and anywhere. What does it mean, anything? When I started using nanopore in 2017 in US, I sequenced E. coli. Yeah. And then after I can sequence the first experiment, after that I feel like I want to sequence everything. Yeah, sequence flowers, sequence leaves, sequence everything that has DNA. So this is mean anything. Yeah. But if we don't have money, we cannot, right? So anyone mean high school student can do that. Professor also can perform sequencing if they can use pipette or if they have credit card to order the machine. And anywhere mean we can do it in Alaska or in the space or even in Thailand, Indonesia, for example. So today I'm going to talk about 4 case studies. What we use nanopore for microbiology world. The first one is SARS-CoV-2 genomic surveillance. The second one is to sequence gene of the bacteria called 16S rRNA sequencing. The third case is metagenomics or sequence any genome. And the last one, again, metagenomics of the degraded DNA. Okay, the first one, SARS-CoV-2 genomic surveillance. This one I collaborate with CDC. Here, we can put the machine into the hood, BSL-2 hood, and to do nanopore sequencing in the hood, like this. But this happened in US before I moved back to Thailand. The director, at that time, it just outbreak happened in US, and people don't know much about this virus. And then they asked me if I can do everything in the hood, not go out, yeah, because we don't know about this virus. That's why it's good that we can put everything in the BSL-2, yeah, and then we sequence SARS-CoV-2 genome in US and also in Thailand. The good thing about this technology is when we start sequencing, after 5 to 10 minutes, we can get the data and perform data analysis. It means that we can detect the infectious disease very quickly compared to other technology that we have to wait for maybe 18 hours or 24 hours and then get the data and perform data analysis. Yeah. And we use nanopore for the first Omicron detection in Thailand. Yeah. It start from getting the sample until getting the result. It takes about 16 hours. After that, we help CDC to do genomic surveillance to detect new strain that come to Thailand. We use nanopore to sequence and provide dashboard back to US, and then they can provide us budget, and then we do sequencing and provide information back to US. The second case is about bacterial identification from amniotic fluid. So in this case, we use 16S rRNA sequencing method. The diagnosis of intra-amniotic infection shown here. The current method, people use gram stain. This is quick and simple, right? But maybe false negative result happen. Also culture. Yeah. This is the gold standard and time consuming. So, and some bacteria cannot be cultured. Then we compare between the gold standard to the nanopore sequencing and also Sanger sequencing. The cultivation take about 2 to 7 days. The Sanger sequencing may be 24 hours to 30 hours. And nanopore sequencing is take about 6 hours. The idea is we get the sample and then we do DNA extraction. You can imagine when we get the sample, in the sample we have human DNA and also bacterial DNA, right? And then we try to enrich the bacterial DNA by amplification, use PCR method, right? And then we load onto the flow cell and throw the data on cloud and get the data that which species. And compare between Sanger and nanopore. You can see here, if we have just one or monomicrobial infection, then the Sanger signal is very good. But if we have like a mixed infection, what happened is blur. The signal of the Sanger is blur. It means that some other technology may be help. That's why if we use nanopore sequencing, even we have 4 or 5 infection or contaminant from the skin, for example, then we can just map to the database and can say that which species happened in the sample. So, and we aim not just for the publication. We ask doctor that what happened if we know the patient got infected with the urea plasma. So we found urea plasma from the sample. And then the doctor said, I can choose treatment that kills this bacteria. Previously, she cannot detect urea plasma because it's not easy to culture. Yeah, in this case, we use DNA base. Then we can identify uncultural bacteria in this case. And we published recently to the international journal. So the next one is, this one I'm a bit excited about this technology. It's called adaptive sampling. This is bioinformatics based host depletion. The idea is when we do nanopore sequencing, and then we will get maybe 400 bases, and then we can map that 400 bases to the target. For example, I can say that if this human DNA, please eject the human DNA out from the nanopore. But if this not the human DNA, please sequence. Yeah, I can program the nanopore to do that. In this case, we got the sample. This is pus sample. And gram stain, you can see here the bacteria, right? And then we do DNA extraction for maybe 50 minutes. Again, in the pus sample, we have DNA of the human, also DNA of the bacteria. After that, we do library preparation using nanopore reagent. It takes about 80 minutes and nanopore sequencing. After 15 minutes of the sequencing, we don't want to wait. Yeah, we go to the machine and download the data from the machine and try to browse, try to map to the database. And we found Piyalitt, but many species based on the browse result, the top hit when we map to the database. But which species is true? So this is what happened in my lab. When we do sequencing, and then bioinformatician and wet lab people come together and see the result. Yeah. And after 6 hours of getting the DNA sample, we do genome assembly. We try to combine all the data and make it like a genome of the bacteria to make it more precise. And what we found Piyalitt BV. And the size of the genome is 2.5 megabases. More important, we found the antimicrobial resistant gene in this bacteria. And we can provide this data to the doctor. And then she can make a decision that, okay, which antibiotic she should use for this case. And this graph for the x-axis show the hours that we do sequencing, and the y-axis show the genome coverage. So in this case, after 2 hours, we get 20x genome coverage. It means that we can make, this data makes us more confident. But if we do sequencing without using this adaptive sampling technology, then it takes maybe longer than 9 hours to get the confidence. Yeah. So in this case, we publish in, for me, it's high impact factor. It's about 10. Yeah, to show the power of adaptive sampling on infectious disease study. Yeah. So in this case, we don't need to do PCR. Just get the sample directly and then do the extraction and then use this technology and get the result within 6 hours. In conclusion, nanopore adaptive sampling can sequence DNA from pus. And now I ask clinician, if they have pus, please come to me. Yeah, we want to learn more and more. Yeah, not just one case. This is just one case, right? And then if, for example, if they have like a pus or some local body fluid, like a joint, from joint, something like that, then maybe we can identify bacteria without PCR amplification. And also, based on this method, we can detect the antimicrobial resistant gene and help physician to make a decision. The last one, case number 4, metagenomics of degraded DNA. In this case, we collaborate with other professor from KU, เกษตรศาสตร์ University. So when I train students, I always tell them, or collaborators, if you want to do nanopore sequencing to get very good quality of the genome, you should give me the sample 1. Sample 1 means high molecular weight DNA. The DNA quality is very good for the sample 1. For the sample 2, it means that you give me the shear DNA, the short DNA. So in this case, the purity is also important for nanopore sequencing. This is, you can use nanodrop. And the size, I expect to see 10 kilobases. And the concentration should be high. However, what I got, so I got the DNA extract from this patient. The DNA quality is very low. Yeah, it's like short read. It's not long read. Right? The quality is about 300 to 500 bases. So my postdoc asked me, should we continue or not? Yeah. However, for this case, I decide to continue to do sequencing. And my collaborator, he thought that this one, he used short read method at the beginning. And he found that, illicit Peyrotrix. But he doesn't know the species. And he believed that this from pig. So he hypothesized that, maybe from animal pathogen. So then he asked me which species if we do nanopore sequencing, and can I detect the antimicrobial resistant genes. In this case, again, we do DNA, we get the DNA extraction and then library preparation. After that, nanopore sequencing. But in this case, we use 2 bioinformatics pipeline. The first one provided by nanopore is called Epitome, and the second one is a Chan-Zuckerberg infectious disease, or CCID. This one is a free software and it's very good. So Epitome said that we got illicit Peyrotrix, but it's not what he expected. Again, we use CCID, throw the data on cloud and get the result back. We get the same result. Sorry. Yeah, we get the same species. 2 bioinformatics methods provide the same species. And then we map this species, the DNA read to the genome, to the this species genome, and we found that it can be this species. And we do genome assembly again, and we found that, this is illicit Peyrotrix, but the species is not the same as what he expected, that from animal. This is from fish. We also detect antimicrobial resistant gene in this case. And we try to find the paper that support our finding. And only one publication show about the fish identification of this species. And also the patient that we that gave us the sample. She also diving for fish before she passed away. So this is very interesting. And in this paper, they use Illumina method, and they found 9 DNA read mapped to this species. But for us, we got genome, and in this case, we plan to publish case report for this case. And now, in my lab, we have different nanopore device. Yeah. And the blue one is coming. Yeah, we hope to use this machine for not only infectious disease, but for host also, to sequence human genome, for example. And this is just example figure that we train students here also use nanopore sequencing. But this photo is from USA. And now we train many countries, so not only in Asia. And this year we plan to go to Italy and UK to train people how to do fungi sequencing. And anyone, if you want to do, you plan to use this technology. So I have like in Thailand, people like Facebook. That's why I create Facebook about the long lead lab. And I will put publications and something that update in Southeast Asia. Yeah. So we collaborate with ONT to provide the information also. But if you don't have Facebook, also I also update about this technology via the Twitter. Or if you have any question, you can message me via the Twitter as well. Oops. Okay, thank you so much. If you have any question, please. Yeah, please. Microphone is better. Yeah.
Thank you. Yeah, thank you very much, Dr.Thidathip Wongsurawat, for the presentation. It's really a nice one to listen to your presentation. I would like to ask, is it possible to differentiate between pathogen and contaminant using sequencing? I mean, maybe from the amount of reads, is it possible? Yeah, that's my first question. Maybe another question I will come to you.
Yeah,
and
sure.
if it's possible for us to visit your lab, it would be very, very nice. Thank you very much.
Yeah, please come to my lab. So, okay. Actually, this is very good question about how to say that this is pathogen if we have maybe million leads, right? So, for example, it's important to have a good pipeline, I mean bioinformatics pipeline, and also database. Yeah, we found that sometimes the database, if they don't have like for example, if the database doesn't have this species, it means that we cannot identify this species, right? So that's why I try to use multiple software and see the consistent between the result. But sometimes to be pathogen or non-pathogen or contaminant, we have to combine clinical data, yeah, and also DNA sequencing result. Because sometimes when we get the sample, maybe contaminant from the skin, yeah. Staphylococcus aureus or some streptococcus can be a contaminant. Yeah. In this case, the clinician will know. Yeah. For this one. Yeah. Yeah, sure.
So the 16S RNA, if I'm not wrong, it's like 1,500 long. So only from the 16S RNA can we detect identify the bacteria, the species?
Yeah. So yes, if we use short read, which is maybe 300, it's hard to identify species level. But in this case, we use long read, right? It's 1,500. Yes, we can identify species from, for example, let me back to this paper. Okay. For this one, we identify 5 species from 5 samples. And some sample we found 4 species. Yeah, in the same sample. Yeah, this is the thing that why we move to long read sequencing. But if you ask me about species, if 3 years ago, I will say no. Because the accuracy of nanopore at that time is not good. Yeah, just recent year, just last year, the accuracy is up and up. That's why now I'm confident to say that we can show species level identification. Yeah.
Maybe next question, sorry. If we want to do like metagenomic from the gut bacteria, because we know that maybe thousands of bacteria exist there, is it also possible to do the 16S RNA sequencing?
Yes, we did 16S rRNA sequencing for elephant dung project, the fecal sample of the Thai elephant. Yeah, and more than 1,000 species. But it's quite hard because no one studied before. Yeah. That's why sometimes unknown species, something like that. Yeah, we use that for comparing between baby elephant and adult elephant. Yeah.
So one flow cell for one sample?
No. So in that case, we use one flow cell for 12 sample. But for pathogen detection, you can maybe 24 samples per one flow cell. Because for gut microbiome, it's more diverse. Then if we combine too many samples, you will get only the top list, right? Yeah, but for pathogen detection from past, from something, then you can combine 24 or even more than that. Yeah.
Thank you very much.
Maybe we can meet around 2 p.m. to visit my lab. Okay. Oh, please. Hi.
Thank you very much, Ajahn Thip. It was very interesting. I was interested about the reads from the degraded samples because because I saw that that quality was so bad and you got only like very short fragments.
Right.
But then you still can perform the long reads. Did you have a special modification maybe in the library preparation or something? Thank you.
Yeah, this is very good question. Actually, Nanopore just released the mode called short fragment sequencing that can sequence above 20 bases. Yeah, they released maybe 1.5 years ago. That's why I think maybe it's matched to that case. So in this case, it's quite hard to why I continue to do sequencing because the collaborator purchased PCR machine for me. That's why I cannot say no. Yeah. However, I learned from this experiment a lot that even this kind of sample we can do that. Short sequence is okay if the DNA has high purity. Purity is very important for nanopore sequencing. If we have dirty DNA like a protein, contaminant or something, it can block the pore. Yeah, but for this case, even low amount of the DNA, but we can still sequence. That's why I put for microbiologists that they can learn from this. Don't throw away the sample. Yeah. No modification, just use ligation technique from nanopore and then sequencing. Yeah. Thank you for the question.
Is there any more questions? We still got 2 minutes for the Q&A.
Okay, two more.
Thank you. If you find a new outbreak which the cause, the etiology, pathogen is not known yet, I mean, can you do pathogen discovery using nanopore?
You mean the novel species,
Yes, novel
right?
species.
Sometimes we sequence and then we can assemble the genome, but it's unknown. Yeah. So in this case, for example, my collaborator, they want to, they have some bacterial species and then they grow and then they publish new species. Yeah. So in this case, it's possible if we can have whole genome. Yeah.
So they use random primers?
We don't need for infectious disease. I'm not sure, but for that case, we just grow the bacteria and then do DNA extraction and then load onto the flow cell. Yeah. Don't need PCR. Yeah.
Thank you.
When we not use the PCR, the good thing is we can study the DNA modification as well using nanopore. Yeah.
Hi, that was an excellent talk, Thip. Thank you so much. So I have two related questions. So one is, now that you have set up this long-read lab with nanopore and you have access to short-read sequencing as well, do you still foresee any use for a PacBio type of equipment? And the second question is then, if you were to plan for any kind of upgrade or additional purchase of equipment that would push the capability of nanopore sequencing further for any applications that you're interested in, what would that equipment be?
Okay, thank you for the question. The first question is about PacBio, right? Actually, each technology I found different advantages. Yeah, even Illumina, nanopore, MGI, PacBio, they have their own good things. Yeah, PacBio, they can give us like a high accurate. Yeah, with 30 kb. Yeah, they give 99.9% accuracy above nanopore. But for infectious disease case, we have to combine many samples, and sometimes we cannot wait, right? Yeah, that's why nanopore may be good answer for the infectious disease. But for 16S, PacBio did very good job. Yeah, high accurate. But it's good for me, I think it's good for research. Yeah, if we combine, for example, soil microbiome, gut microbiome, many samples, and do research, I think if I can access technology, maybe I choose the PacBio. Yeah, it depends which technology I can access. And also MGI is also good, yeah, for short read sequencing and cheaper compared to Illumina. Yeah. However, now when I have to do short read, I send the sample out to China. It's cheaper. Yeah. Yeah. So I think depends on the technology that I can access. And can we wait to combine multiple samples at a time? Yeah, this is what I think. And then I asked my team, if people gave us PacBio 10x, do they want? They say, oh, even nanopore is too many works, something like that. Yeah, that's why, okay, let's see if we have more space or more people, then we will have more technologies. Yeah.
Thank you.
And another question about the future technology, right? Actually, for me, I try to combine other technology as well. For example, twist bioscience, like a probe capture, not just nanopore sequencing, but can we do something like capture some organism, virus, for example. I did not show here, but we try that. We try to capture only the virus, 3,000 possible virus using twist bioscience probe. Yeah, we try to combine many technology, to help to enrich the bacteria, virus, plasmodium, something like that. Thank you for the question. Okay.
There will be a sharing session after the conference, so if you guys have any more questions for Dr. Thidathip, please hold on to your questions. Thank you.
Thank you so much, Dr. Thidathip. So now we would like to invite Dr. Chayasith Uttamapinant from School of Biomolecular Science and Engineering, Ittichaiwong Methi Institute of Science and Technology. Everyone, please give him a warm welcome.
Thank you so much for your kind introduction, and I hope you can hear me okay. All right. All right, so we actually have 3 talks today, and as Ajahn has told you, she has talked extensively about technologies that can read DNA, genetic sequences. And for the last talk from Dr. Piyalitt, you would hear about how you can design kind of even new technologies using artificial intelligence. So for my part, it's actually the middle talk, the middle child. I'll be talking about how we can write and edit genetic sequences based on the information that you get from reading and also from the design. And the focus would be on CRISPR-based technologies that you can use to manage infectious disease in many ways. And then, so where I work is a new university called WISTEC. I'm actually not part of the Siriraj hospital staff, but we do collaborate extensively with Siriraj here. And this is actually my campus. Unfortunately, you cannot visit my lab because we are based in Rayong. We are close to 3 hours from here by car. And this is how the campus looks like. It's in the middle of a jungle, just with a few buildings. So it's really nice and calm in a different way compared to Siriraj. And the department that I work with is Biomolecular Science and Engineering. We actually are quite small. We only have 6 faculty members, but the focus of the research in our department is really quite specific in that we are developing biotechnologies that hopefully would have applications both for bioindustry and also for medicine. And this is particularly through the use of high-throughput enzyme engineering technologies and directed evolution, development of biosensors, as well as cells. And the project that I'll be describing today is a project that we have been working in extensive collaboration with Siriraj Hospital. So the 6 researchers at the top are staff from here and also from my team at WISTEC who have been developing CRISPR-based technologies for infectious disease management. And the projects were done in close supervision with Professor Nawin Hotongkang at the Department of Microbiology here, and also Professor Danaya Pakotipapa at the Department of Biochemistry of Mahidol University. So with CRISPR, what it is, I actually think that the audience know this already, but I'll just give a quick recap in that as the topic of this conference is, trends within the microbiological world, CRISPR is actually the prime example of a gift that the world of microbiology has provided to us biotechnologists. So what it is basically an adaptive immune system that exists within bacteria and also certain viruses that allow these bacteria to fight against the parasite, in this case the viruses. So when you have invading viruses, which has genomes, may it be DNA or RNA, the bacteria can then remember these foreign parasitic sequences and then kind of copy that in the form of a CRISPR array And then after that, the bacteria can then form what is called the interference complex, basically the sequence of the foreign DNA or RNA being in complex with a really cool enzyme called Cas or CRISPR-associated enzymes. This is an enzyme that when it is being programmed with this foreign sequence, would then have a really high specificity towards that sequence. So once that bacteria cell encounter the same virus again, it would know that this is the enemy, and then it would home that destructive enzyme is the nuclease onto the virus genetic information and then clip that off. So this is the way that nature has basically evolved a really cool adaptive immune system to fight against viruses. And this is the work that then has been repurposed extensively within the biotechnology field, particularly for gene and genome editing. And the work was pioneered by these three scientists, Jennifer Doudna, Feng Zhang, and Emmanuel Charpentier. And two out of the three researchers, Professor Doudna and Charpentier, won the Nobel Prize in Chemistry 3 years ago. And the reason why this technology has become a hallmark of how we write DNA is that it is so easy to use. You basically can almost edit any sequence just by designing the guide RNA or the CRISPR RNA molecule that is the programming, basically the memory part of this whole CRISPR system. You can design that super easily and then really just home in these Cas enzymes to do almost any function that you want, may it be just cutting or even inserting a new sequence within DNA or RNA. And the ability to do such editing actually has far-reaching applications. So this is more like a sci-fi in a sense, in that you can dream that one day you can make organisms almost on demand. If you have tools that can fully customize the genetic markup of that organism, you can imagine making like this kind of web-based interface where you select the properties of the organism and then tailor the genetic sequence to be exactly for that. And CRISPR indeed has been using as a basis to generate such drastically altered organisms. So this is one example where you can do full genetic recording of a bacteria called Escherichia coli. So this is, Dr.Thidathip just mentioned that the size of the genome for an E. coli bacterium is around 5 million base pairs. So now there is a way to fully change those 5 million base pairs of the genome so that it becomes something completely different. And one thing that people in the field of synthetic biology are interested about is how can you maximize the utility of the genetic information that are stored within those 5 million base pairs. This is something that you learn from classic basic biochemistry class in that you have 20 amino acids that are being encoded actually by 64 codons. So basically 3 nucleotides that are giving signal to encode a given amino acid. But you will see that there is something that is kind of funky with this number in that why would you need 64 alphabets to encode 20 readouts? And then alternatively, can you actually use these 64 alphabets to encode 64 readouts so that you can fully maximize the types of side chains and functionalities that may exist within proteins? So with this, there is now a way to kind of probe this question by fully edit the genetic code of a bacterial cell. This is the technology that I work on as a postdoc in the UK. Is what is called REXA. So it stands for Replicon Excision Enhanced Recombination. The name sounds a bit complicated, and there is actually quite complicated scheme. But all you need to know about this technology is that it allows you to cut and paste really large sequence of DNA that you chemically synthesize in a test tube. But you want to then get this chemically synthesized sequence and then put it on the genome of a bacteria precisely. And by doing this precision swapping, this is what we use CRISPR-Cas9 for. And with this, we create what is called Sin61. So it's an E. coli cell that doesn't use 64 codons anymore. It only uses 61.
And then the cell is actually fully viable. It actually looks just like normal bacteria. But now what happens is that we free up 3 codons out of the 64 so that it can be used to encode completely new building blocks of the amino acid. So this is the work that I think the field of synthetic biology is really excited about. And then with CRISPR, we talk about making kind of drastic changes to the whole genomes. But then when it comes to human health, there are also clear applications that it can be used for. And it has received intense interest as a tool to develop, to be developed as therapy, especially in humans. So there are many modalities that you can do such gene therapy with CRISPR. So you can take cells out of the patients, maybe stem cells, and then you do editing based on CRISPR ex vivo, so outside of the patient body, and then you transplant these cells back into the body, and then allow them to differentiate and then replace the faulty cells. So this is the approach that is now being pursued quite a lot in the first phase of gene therapy based on CRISPR. And you will see that within the past few years, there are extensive clinical trials that use CRISPR as a basis for gene therapy for many diseases. And this is the news that I'm sure that many of you have seen recently, that the first gene therapy that is based on CRISPR has been FDA approved by both the US and the UK. And this is based on the use of Cas9 to edit a gene that is called BCL11A, which normally suppresses the expression of one form of hemoglobin that is so good at carrying oxygen. And then in the patient that has sickle cell anemia, this form of hemoglobin is not being produced anymore. But then once you cut this BCL11 gene, you then release the suppression and then allow the hemoglobin to be produced. So this is a really, really cool technology where you specifically only inactivate one gene, and then you fully reverse the phenotype and then allow the patient to hopefully be cured. So that's one example. You can also use CRISPR as a way to fight not only genetic diseases, but also invading pathogens. So you can use CRISPR as antimicrobials, basically turning its own tool against itself. So in this case, you can package the CRISPR-Cas machinery within viruses, for example, phages, and then you can then allow the phage to infect the bacteria, which may be the pathogens. And then because bacteria is not really that good at repairing its own DNA through certain mechanisms, when you have too much of these cutting machineries within the cell, it effectively would act like antibiotics and would kill the bacteria. So this is technologies that are under intensive development throughout the world. And the work that most people are familiar with, the work that is based on gene editing, and this is mainly done with an enzyme called Cas9. But there are actually many diverse types of CRISPR-Cas systems that act on double-strand DNA, single-strand DNA, and also RNA. And these are the types of enzymes that my lab is actually using quite a lot, not only for editing applications, but for genetic detection applications. And this is the part where I will talk about our research in collaboration with Siriraj. So we have been working to develop a CRISPR-based detection system or diagnostics, and also generating resources such that the research and also the implementation could be sustained within Thailand and Southeast Asia. So we have a few publications within this pipeline already, and also one of the first FDA-approved CRISPR-based tests for COVID-19 for Thailand. So with CRISPR-based diagnostic, it's almost the same concept as what I described to you before for gene editing, except that in this case, you are using the CRISPR system to detect as opposed to edit. So within a diagnostic platforms, you can have a combinations of technologies within it. On one hand, you have techniques that can amplify and increase the amount of genetic materials within the sample, and this allows you to have a highly sensitive detection technologies. The class of amplification technologies that we love is isothermal amplification. So a reaction that can increase the amount of DNA at a single temperature so that you can perform this reaction easily within the field without having to use a thermocycler. And on the other hand, on the right side, once you amplify the genetic target, you can then design the CRISPR-Cas system such that it detects that genetic target in a specific manner. And the types of Cas enzymes that we love to use are Cas12 and Cas13. So these are enzymes that target either DNA for Cas12 or RNA for Cas13. What is cool about these two enzymes is that once it recognizes the target, it not only cleaves the target, but triggers kind of a cascade reaction that allows one detection event to be amplified into millions of cutting reactions. So this principle of signal amplification is perfect for detection applications, meaning that you may only have one copy of the target, but you end up having millions or 10 millions of reactions that you can generate to give you enough signal for the detection. And this is the work that we have been doing in Thailand for the past 3 years, so combining these two technologies to develop diagnostic applications. And there is a reaction that we love to use for the amplification. It's called RPA, stands for recombinase polymerase amplification. And then we highly love Cas13 enzymes for the following reasons: that both RPA and Cas13 are really good at doing multiplex and also working at near ambient temperatures. So in between March and May of 2020, we work extensively with Siriraj Hospital and actually do one of the first large-scale clinical validation of a CRISPR-based test for COVID-19 in the world. And by May of 2020, we actually were helping patients who are going into operation here at Siriraj to do a test for COVID based on CRISPR. And even at that time, we know that the technologies are working really well. It has really high specificity and sensitivity, but we also know that it's not perfect in that the technique itself involves a lot of manual handling time. It involves using space segregation such that maybe it's not that useful when you want to use at point of care outside of labs. So from that, we have been working to really improve the technologies and then hopefully extend the testing to a location that is now decentralized. So in terms of target product profiles, this is what we are aiming for in terms of the development. The test has to be really easy to use. The time to result has to be really quick, ideally within an hour. You don't need expertise to interpret any results. Hand-on times has to be quick, and basically all these conditions. And most importantly, it has to be cheap if you want it to be adapted. And our angle to get the technologies to be cheap is that we want a given reaction to be as useful as possible. As Ajahn Thip mentioned, that a given flow cell, ideally you want to use it for 10 or 20 samples. In this case, it's the same principle in that we want one biochemical reaction that we are running, even though it's just 1 microliter. We want that to detect hopefully 10s or 20s of pathogens at the same time. And with this kind of multiplex detection, it actually needs a lot of innovation from biochemistry, from device design, from probe design, and also from resource analysis. So this is what we are working as a large group together right now, particularly with Siriraj. And one way, one example that we have already demonstrated the utility of this multiplex detection is by doing basically SARS-CoV-2 genotyping. So basically one detection reaction that not only tells you that you have a COVID-19 infection, but tells you almost immediately that you have which specific variant of concern that may be infecting you. And with this reaction, it's actually quite simple in that instead of just detecting one genetic target, you are detecting 2 or 3 genetic targets using multiplex recombinase polymerase amplification, followed by multiplex Cas-based detection using orthogonal Cas enzymes. So these are different Cas enzymes that have different kind of programming capability and also cleaves different sequences. And then you can basically program the cleavage of the Cas enzyme such that it gives off a different fluorescent color. So in this case, if you have pathogen 1, for example, it will give you a green color. when you have pathogen 2, it would give a red color, and when you have both pathogens, you have the mix of green and red, which is yellow. And in terms of developing the protocols into something that can be used at point of care, as mentioned before, if you have multiple components of the reaction, it's annoying to use. So in this case, we pre-mix everything into one tube. And not only that, we develop a formulation that allows freeze drying of the reaction so that they can be easily transported and also used at the point of care. So you basically have this freeze-dried pellet. You just add buffer or the RNA or DNA solution, and you basically allow the reaction to proceed immediately. So with this, as mentioned, we have developed the test as a way to do genomic surveillance of SARS-CoV-2. You can differentiate between, at that time, different VOC variant of concerns all the way to Omicron quite specifically, and also with high sensitivity. So beyond COVID, we are now working on developing the test for multiple infectious disease. And one example that I'm giving today is a point-of-care CRISPR-based test for myelioidosis. And this is work that we are doing in collaboration with Dr. Chayasith at Mahidol Oxford Research Unit and Dr. Somsakul Wongpali at Chiang Mai University. So myelioidosis is a highly deadly bacterial infection caused by Burkholderia pseudomallei. It's highly endemic in the northeastern part of Thailand. And Claire and Pop ran a clinical study for the past 3 years. And the statistics is super depressing in that we are seeing high fatality rates of patients who basically pass away even before they receive any diagnosis results. And with myelioidosis, it encounters the same issue with many infectious diseases in that there is a right drug for it, but unless you have the right diagnosis, there is almost no way that you can kind of foresee and then give the correct drug. So having an early and correct diagnosis, ideally within 1 week of the symptoms showing, are already known to be super helpful to the patients. It reduces the fatality rates to be down than 10% as opposed to now 50%. But then the gold standard of doing myelioidosis diagnosis is using culture, which normally takes a week. So by then it's too late. So we have been working to develop a point-of-care CRISPR-based test for myelioidosis, and now we have shown that the test actually has sufficient sensitivity not only to detect the bacteria that has been cultured, but it can also be used to detect the direct samples in pus, sputum, and in urine. And this greatly facilitates the diagnosis where you basically eliminate the time to do culture immediately. And then when you analyze the time to results that you obtain from a CRISPR-based test of myelioidosis, basically it now takes hours as opposed to days. And this allows the correct antibiotics to be administered. And then aside from the work on different infectious diseases, my team is we have a lot of biochemists. So we have been creating resources of reagents that can be shared among researchers and also be used as a basis even for clinically approved tests. So one of the reagents that we have been working quite hard on is the reagent RPA, the isotope application, which is an annoying reagent in that you can only buy it from 2 companies in the world. And sometimes it takes a few months before you can get the reagent from the companies. So we have been trying to crack the recipe for this reagent, and we have succeeded in that we can now produce this reagent RPA, which is quite complex. It has many proteins and many cofactors in it, but we can now formulate the reaction and then demonstrate that it has equivalent efficiency to the versions that you buy. And we hope that this would allow uptake of these point-of-care tests, hopefully in the future. And the recipe has already been reported. So moving forward, we are still working really closely with Siriraj, where on our side at RISTech, we are developing prototypes of the test and also improving a lot of components within the test. RISTech is not a hospital, so we are relying on the technical expertise on the clinical side of Siriraj. So you can foresee the synergy and extensive collaboration between our 2 universities. And then this is one example where we push this collaboration all the way to implementation, where we have an FDA-approved test for CRISPR, test for COVID for Thailand. And then in terms of capacity building, we now have a yearly class at Siriraj where we teach point-of-care innovative technologies, including CRISPR. So this is run in conjunction with the Department of Microbiology here. So if this is something that our colleagues at other universities, not only in Thailand, but also from Southeast Asia and other countries are interested, please feel free to get in touch. We are happy to share any resources for teaching on this. We also hold a workshop late last year on CRISPR-based diagnostic that also have researchers from Southeast Asia that coming to train at WISTEC for 3 days. And this is something that we hope can help not only infectious disease management in Thailand, but also within the region. And obviously, there are many other diseases that are endemic to our region. So we don't have hands to do everything right now, and it actually would be really cool that universities across the countries can work together to develop technologies like this. And with that, I would like to conclude by thanking the people who are doing the work, and also the work on CRISPR has many more contributors beyond the faces shown in the previous slide. So researchers at WISTEC, at Siriraj, and also at Mahidol Chulalongkorn, and also the Ministry of Public Health, and also international collaborators to the project. And lastly, my group and also funding. I'll take any questions now. Thank you.
Now it's time for the Q&A session. If there are any questions, please feel free to raise your hands and we'll give you the microphone. Thank you. Okay, there is one question over here.
Excited about RPA that you mentioned only 2 companies can do that, and you try to crack, right? And you plan to sell like a WISTEC brand, something like that?
So the ability to manufacture a reagent like that actually doesn't just allow us to make it for commercial use, but we actually can engineer it to make it even better. So that's actually an ongoing research in our lab. And we do have that improved formulation with an engineered component already. And ultimately, that is something that we have been discussing with the ministry. So we hope to scale the production up, but we want it to be also widely accessible. So we are trying to find ways to scale up the production while keeping the cost down. And that has been quite challenging. Let's put it this way. But we are trying to do that right now.
So I hope to see Thai brand. Yeah. Thank you so much.
And that's actually, there is a related kind of question or point to that in that technologies like this are also quite tightly tied by intellectual properties, by patents. So we can make it and then use it even for commercial uses in Thailand because, for example, patents for RPA are not filed in Thailand. But to extend the technologies potentially for use in other countries, there may need to be even further extensive engineering to the formulation such that it's not being tied by IPs anymore. So it's kind of tricky in a way, but you would want to extend the use of the technologies to be as wide as possible. So we're going there.
Are there any more questions?
Okay, so thank you, Dr. Chayasith, for. Oh, there's one question.
Thank you. Dr. Chayasith Uttamapinant, thank you very much for your presentation. I'm lost in words, actually. Very excellent work from you and your group. I forget my question already. Yeah.
We can talk later. Don't worry at all. Yes.
Yeah, I was wondering how can you get molecular techniques and put it in lateral flow form?
Right. So that was actually our original work in that we are linking this CRISPR-based detection and then linking that to the lateral flow strip. So with that, there is a way to design the reporter for the Cas enzyme such that it can trap basically antibodies conjugated to gold nanoparticles, and then that would give color on the lateral flow strip. We kind of move away from that just a little bit because the strip version of the test has lower sensitivity compared to when you use fluorescence. But we also know that the advantage of the strip being fully kind of field deployable cannot be beat. So it's kind of knowing now that the strip has maybe reduced sensitivity, we are then improving the sensitivity of the reaction itself by doing enzyme engineering and so on, so that it can offset that lower sensitivity from the strip readout. So we are working on that too.
Great, thank you.
Okay, so thank you so much, Dr. Chayasith, for the wonderful lecture about CRISPR technology for diseases, infectious diseases, management and diagnosis. So the next session will be the online session from Dr. Piyalitt Ittichaiwong. We will meet him via online meeting. So is the Zoom meeting ready? Okay.
Sorry, do you hear me?
Yes, I can hear you very well.
Okay. That's clear. Okay. Sorry, everyone. The sound is good, right?
Yeah, it's perfect.
Okay, thank you. Okay, sorry that I cannot join at the Bangkok because now I'm at the London. Okay, today I will talk about AI in the microbiology and parasitology, but I will talk in the broad picture about the AI. Yeah, because the AI not only can help in the microbiology and parasitology, but it can also help in the many tasks in medicine. Okay, first, my name is Ittichaiwong. Now I'm working at the Siriraj Informatics and Data Innovation Center, or you can call it SIMPIC. And now we're working focusing on the two things. First, we focus about the data, and the second, about the data management, and the second is about the medical AI. Yeah, and I'm very grateful to talk today. Okay, but okay, sorry, I think it's my connection. I will close the camera because I think my connection is not good. Okay. Okay, and next, I will talk about the conflict of interest first. I am the co-founder of Preceptor AI, a startup where we are pioneering in developing language models akin to ChatGPT. Our specialization lies in tailoring these models for medical professionals in the ASEAN region, utilizing exclusive medical information to fine-tune our AI. aiming to reduce the rate of erroneous outputs, commonly known as hallucinations. Additionally, I co-founded Sense AI, a company at the forefront of wearable device AI technology. We focus on developing AI capable of detecting abnormalities in human health. A prime example. I'm so sorry, I think the skin is not sure why. Okay, I will try again. Sorry. Yeah, do you see it? Oh, oh, sorry. I think I chatted the screen now. Yeah. Um, slowly. Okay. Can you see my slide now? Okay, I'm not sure if you cannot see.
The
Okay.
slide hasn't showed yet.
Oh no. Okay, I think it's maybe because the internet connection at London. Okay, but okay, so I think sorry everyone. I think maybe it would be better if I post the video clip in the page. Yeah, but now I try to post the to share the screen. But if it not work, I think yeah okay I will record the video and send to the SIMPIC page. So okay so in short I'm sorry for everyone yeah because I'm not think we will have the internet connection now but I think it's from my side but okay I will send the video to post in the SIMPIC about my part but if in case if you have a question you can ask me at my Facebook or my email that I will post it in the my video clip and I'm really sorry about that. Sorry. Yeah, but I'm not sure. In my screen, I see my screen, but you don't see my screen, right? Oh no.
No, we can't see your screen, but maybe we try to open your clip and then the live Q&A session via the Google Meet. Is that possible?
Oh, yeah, but okay, but yeah, but I think the video clip is still uploading now. Yeah, so I think it may be not available in time. Yeah, so oh wait a minute, I will change the system. I'm so sorry everyone. Um okay but okay. I think I changed the user. Okay, can you hear me?
Yes, we can hear you very well.
I'm not sure. Do you see my slide?
Yeah, we've seen your slide very perfectly. Thank you so much.
Okay. Then thanks so much, and sorry, everyone. Okay, and for me, as I talk, that first I need to declare about the. Oh, wait a minute. Okay, I need to declare about the conflict of interest because I am the co-founder at the piece of the AI. There is a startup that focuses on create the large language model that, but it focuses fully on the medical field. Yeah. Yeah, and the second startup that's called the SeniAI that we develop AI for the wearable device at the Samsung Watch or the Apple Watch to help for the diagnosis of the obstructive sleep apnea. And the third is I am the part of the lab team at the OpenAI. Yeah, so because today we talk about the GPT-4, so I need to recap first that I will. Okay, I think that they will effect. Oh. Hello, 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 hello. Oh, 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 oh. Okay, 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 okay. I'm not sure why now there is a problem. Oh, 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 oh. Yeah, so sorry, I think it's, I think I will send to post the clip. I think it will be better. I'm sorry, I'm not sure what happened now. So sorry, everyone.
Sorry. Sorry, I think there is an echo in your microphone because you have two devices in the meeting. So maybe you um, leave one device from the meeting, if that's possible. Okay, so due to the technical problem, so we will switch the sharing session of Dr. Thidathip and Dr. Chayasith instead of the online meeting of Dr. Piyalitt. So next will be the sharing session, and we will follow and the online session, the online video clip of Dr. Piyalitt will follow after the sharing session. So we will now begin the sharing session.
All participants can stand up from their seats and move down to the near the stage area to ask Dr. Chayasith any questions. Dr. Chayasith is still with us today, and I am really inspired by his talk because I have came across the term CRISPR before in my high school biology class, and getting to listen today was really clarify what the term means and also the hope for future healthcare of Thailand and how CRISPR can really diagnose and be very beneficial for patients.
Me too, as a second-year medical student, I feel very inspired by his talk. So every participants are welcome to join the sharing session with us today.
We have about 20 minutes left.
Okay, so for the participants via Facebook Live, this is the end of the Facebook Live now. So thank you very much for participating our Facebook Live. And for the participants in the Rashtrapanada meeting hall, if you don't have any questions, you can feel free to leave the meeting hall. Thank you so much.
Thank you for coming today.
Once again, we are deeply sorry for the technical problems, and we feel so thankful for every participant in this room and in the Facebook Live. Thank you.